character-based tree reconstruction. And there I introduced you to the idea of small parsimony. So I'm basically going to repeat that a bit, revise that. So the idea is that you have a multiple sequence alignment of n species, and this alignment is based on m characters. Now what you essentially have to do is, you based on these m characters, you have to find the ancestral sequence of the internal nodes and the root node. All right, given a tree. So basically this is what you want to go. You want to go from a tree with made from the sequences that you've been given to a tree with the internal nodes also being assigned with a particular sequence. And why do you do this? You do this essentially to ensure or understand exactly how evolution is proceeding. That is what is the best way in which, or the simplest path, the greediest path that the evolution is taking down this into like to obtain species currently that you have. Although I say it's greedy path, but you will see that it's actually it might it might be different also, right? It's not greedy algorithm will not give you the right answer always. So in order to do this, in order to sort of ascertain or confirm that the assignment of internal nodes that you have is ideal, what you do is you calculate a Hamming distance score from the internal nodes to the leaves, right? So for example, here I'm showing you the Hamming distances. Chimp is different in one place, human is different in two other places, right? So you basically have Hamming distance of 2. In same case here, the seal is different in two places, whale is not different in any place. So the Hamming distance is 0. And now if you go compare this with the root node, there is a difference of 1 as in the green base, and there's a difference of 2 in the orange bases, as you can see. Okay, and the idea is to calculate this parsimony score, which is the sum of Hamming distances along all the edges, and the best tree is going to have the minimum parsimony score of the assignment that you're obtaining. All right. So the small parsimony problem is defined as the most parsimonious labeling of internal nodes of a rooted tree. What do you mean by most parsimonious? The given rooted binary tree with each leaf labeled with a string of length m. The output is a labeling of all the other nodes, which is internal nodes of the tree, such that it minimizes the tree's parsimony score. So the most parsimonious labeling means minimal parsimony score. Now, if you want to simplify this problem, what we can do is, if we can solve this problem for a single symbol, then we can extend this problem across multiple symbols. All right. So what do I mean by a single symbol? So earlier I was showing you the entire sequence, but now let us focus on just the fourth column here between all the sequences, and you try to build a tree. So how do you do it? So basically the idea is that for two leaves, right, the most parsimonious score can be determined like in a sort of a greedy way, where you say that, okay, the best score that is assigned here, right, or the best sequence that is assigned here, I'll assign a sequence here which gives me the best result, right, or the minimum score for these two things. Similarly, if you go up the tree, the best score for this, or the best parsimonious assignment for this node, is going to be the best between this and this particular subtree. All right. So TB here denotes the subtree of tree T, where whose root is V, right? So TV is the subtree with the root as which is originating from the node V. So we basically define something called score SK of V as the minimum parsimony score of TV, this subtree, over all the labelings of TV. All right, assuming that V is labeled by K. All right, is this clear? So the minimum score for this is defined as the minimum score obtained from all combinations of TV, right? And the other node that it is coming from. Okay, so the minimum parsimony score for the tree is equal to the minimum score value of the SK root over all symbols of SK, over all symbols of K. So I'll show you how this works. So let us see, let us say we have these letters, right? And in the leaf. Okay. Now let us go at this end. So this is a T, this is a C, right? So now if you assign this node A, what will be the score? If you assign this node as A, what will be the score? 1 and 1, right? You'll have to make one mutation. Is everybody following me?
Sir.
Right? Similarly, if you put this node as C, you'll have this would be length 0, this would be length 1, right? If you put G, this will be again 1, 1. If you put T, this would be 0, this will be 1. All right. So just like that, at this particular internal node, we calculate what are the possible assignments, right? And the values for those assignments. So remember, in dynamic programming algorithm, we basically, in order to reach the final path, we calculate the best assignments for each of the individual paths. All right. So here, what is going to be the best assignment for this particular node?
ACGT.
Can be C or can be T, right?
Yes, sir.
So we are only dealing with one letter right now. Okay.
Yes, yes.
Here, what will be the the best assignment?
G.
This can be either A or C, and this can be again C, right?
Yes.
So now let us make those assignments. And now let us go back. Let us go further up to this particular node. Let us say this was an A, right? If this was an A, then because this is definitely going to be a G, this length becomes one more, right? So this parsimony score will be 1 like this. And if this is an A, right? Because this is C or T, so you're going to add one more here, and there was anyways one difference between this and this here, one more. So the score for A becomes 3. If you have C, then you can calculate the scores, right? With G, you can calculate the scores, and T, you can calculate the scores, right? So like this, you keep on building up and up. You find at each point using uh, this sort of optimization equation, that for all symbols I, so for all symbols ATGC here, what is the best score for the daughter, that is T, right? Plus the son, right? Given you calculate this delta IK and delta JK, so this is I and this is J. So given you calculate the score based on the assignment of the symbol I to the sort of the root node here. Is this clear? So this is how the algorithm looks like. So for all symbols I and J, define delta Ij equal to 0 if Ij equal to J, 1 if it's otherwise, right? And this is the optimization that you're doing at each node. All right, so finally you assign C here. When you assign C, you will have minimum number of mutations along the tree in order to get the most parsimonious tree. So can you now backtrace, given I solve that this is a C here, what is going to be the assignment?
What does it mean? Like, what are you saying? Assignment?
So this is, we have reached the sink, right? So this was the source.
Yes.
Remember the root problem, and this is the sink. So we have reached the sink. We know that in the sink, C is the best assignment, right? C is the best letter. Can you assign the intermediate nodes now?
Sir, can you read why the second node in right has 3 in A?
Yes.
It should be 4, I think.
It should be 3, right? If it's A, let us say this was A, right? And this was G. So there is going to be one change here and there is no change here, right? So this value is 0 here. If this was A and this was C or T, there's going to be one change here, right? And because this was C or T, there was one change here, either ways. So the value is going to be 1 plus 1 plus 1. That is 3.
But we do not know what is the value in these intermediate nodes. So are we assuming based on the minimum score or?
Yes. So you get the minimum score for each of the nodes, right? And then
Yes.
you get to the next node and you see based on the minimum score of each of the nodes, what is going to be the value,
Oh,
minimum value for this. So that's the idea, right? So let us say we start from the bottom, right? Here, there is nothing, there is no value here, right? At the bottom. So we basically ate everything which is not this letter is basically infinity. So this letter is equal to itself, so it's 0. Now we go here. Let's say we compare this node right here, right? It's GG, so this one has to be G, right?
yes, sir.
And so this is already assigned as G. This one can be C or a T. We don't care. So let us say it is a C, right? Then here it can be a C, a T, or a G. All three possibilities are working here. All right. So do you want to fill with me? So what would be here? Here is G, right? Definitely. So
Yes,
what will you fill here? Will you
CO.
fill a C here or a T here? Given that this is a C, you will want to minimize this, right? So you will fill a C here and you will fill a C here. I'm not explaining it a little bit. I mean, as much as I was explaining earlier, because I was hoping that guys will be able to follow this. Is this clear or not?
So it's clear.
Sir, it's clear. Yes.
Right? Once you have calculated these things, now when you're backtracing, you go from the root node down and you see, okay, if this is assigned as C, which should I assign it? I don't want to assign it A because then there'll be a mutation. I don't want to assign it G because there'll be a mutation. Don't want to assign it T, right? You will choose the ones which are minimum here. Out of the minimum one, you take the one which reduces the difference. So you take the C. Now you go back down here. There is C and a T here, right? Which are the minimums. So you choose that one which reduces the difference having distance. So you assign it as C. So here again, this was C. Now when you go down here, the minimum is G. So there is no other option but to take G here. Let's go to this one. If we go here, the minimum is C, so that choice is already made for you. When you go down here, it can be either A or a C. If you take A, you add one mutation. If you take C, there is no mutation added, right? So you decide this to be C. Where it's minimum, it's quite straightforward. So please code this. Write a pseudocode for this. All right. So again, the small parsimony problem in an unrooted tree is basically find the most parsimonious labeling of internal nodes of an unrooted tree, an unrooted binary tree with each leaf labeled by a string of length m. A position, the output would be the position of the root and a labeling of all the other nodes of the tree by strings of length m that minimizes that tree's parsimony. So earlier we were talking about a rooted tree problem. The same thing extends to an unrooted tree as well. Just that here you'll have to find both the position of the root and labeling of all the internal nodes. All right. Now this can be extended to what's called the large parsimony problem. So what do I mean by large parsimony problem? So for example, here, when I was telling you that we have to find the most parsimonious assignment, I was already giving you the tree, right? I was telling you that chimp, human, seal, and whale, for example, are organized in this tree that this is the best distance based tree here. All right. And then you get the parsimonious assignment of the sequences in the internal nodes. So that problem is solved. But what can happen is you can actually extend this to a problem where you are trying to maximize, also optimize for the tree itself, right? So the tree is not fixed, and you also want to get the best tree given, which gives you better chances of a better parsimony. All right. So you try all possible combinations, and you then for each possible combination, you try to assign the most parsimonious sequence. So what is large parsimony problem? Given a set of strings, find a tree first with leaves labeled with as with all the strings having the minimum parsimony score. So input will be a collection of strings of equal length here. When you say equal length, you essentially will have an alignment, right? And the output is a rooted binary tree that minimizes the parsimony score amongst all possible rooted binary trees. All right. So you not only have to calculate the parsimony score here using the small parsimony algorithm, but you also have to find all the possible permutations of the tree leaves, right? To get the best tree. This is because you have to find out all possible trees. This is unfortunately an NP complete problem, so it takes a long time because of finding all the possible trees out of n possible strings. This is basically is exponential in nature. So this is the greedy algorithm, so to say. This is optional. That's why I've written. I would not be asking questions about this, but if you are interested, how you would do it, you would set the current tree equal to the arbitrary binary rooted tree structure. You will go through all the internal nodes, perform the possible nearest neighbor interchanges. You'll solve the small parsimony problem for each tree. If any tree has a parsimony score improving over the optimal tree, you set that tree to the current tree, right? So here you have to basically go through all possible trees, right? There is no way of actually prioritizing which tree is going to give you a better parsimony score. All right. So this essentially means that distance-based trees are not always very accurate in terms of defining internal states. They might capture evolution, but they might not actually be able to capture how the strings are changing that efficiently for the internal nodes. All right, is this clear? So I want you
So
to be able
I have
to,
a doubt here.
yeah.
So how is this approach different from the previous one? Because there also we were checking all possible trees, right?
But
This is that approach only. I mean, I'm just showing
Okay.
you one greedy way of solving this, that you take one tree, you basically arbitrarily go through each tree, you interchange the internal nodes, right? Basically, you the small parsimony problem for each tree. And if your parsimony score is better, right, then that becomes your optimal tree. And you keep on doing this until you have gone through all the trees. So this is just the greedy way of solving this problem. Yeah. Here I was
Yeah.
just stating the problem. The simplest way of solving the problem, right? You just go, you solve for all possible trees. That's it. Ratnesh, you had a question?
no, sir, no.
Okay, so now how do you actually use these evolutionary trees? What can you do? So one of the first things that people used this was to figure out exactly how humans have evolved. And here there was the first this first kind of trees that were made for genetic analysis was made using the hemoglobin gene, because hemoglobin gene was the first genes which was sequenced. Blood was easy to also hemoglobin was easy to extract from blood, so you could also get the protein sequence of these genes. So uh, this was the idea that was put forth, that the gorilla hemoglobin seems to be as abnormal as the human hemoglobin, suggesting that gorillas and humans would be close to each other. But this was something that was sort of not accepted by scientists at that point of time. But this is in fact true. But it gives you an idea that was put forth at that point of time was that the choice of the molecule that you're looking at determines what similarities you will be seeing. What do I mean by that? I'm going to explain to you slightly later. So the other things that it was used to do, just like for humans, we were trying to understand how humans evolved. People were trying to do this for a species like panda. So they were making these evolutionary trees, and you could basically trace how many years ago panda was evolved. And that's how a lot of countries are sort of claiming that a particular animal or a particular plant, which is medicinally important, it was sort of indigenous to a specific area of the world based on millions of years ago. It was found, and when you look at all the plants that are related to that plant, should also be closely found in the same geographical area, and also should show similar uh, time point at which the evolution happened. Uh, we can do this for humans as well, trying to see at what point of time you had like how do humans around the world relate to each other. So the reason why we say that most humans have come out of Africa is because if you basically make a tree out of the genome, specifically in this case it's the mitochondrial genome of all the human beings which are there with respect to Africans, you see that we are actually clustering together with a sub branch of Africans. You can see here the Yoruba tribe, for example, which was supposedly the people from uh, this tribe went out of Africa and colonized different parts of the world. And that's how you get this kinds of maps where you say that, okay, first people were inhabiting in this region. When they moved, the people, if you take genetic samples from different regions of the world, you see that based on the phylogenetic clustering, at exactly what time point these clusters appear, right? How does the trees look like? And now when you go from the root to the ancestral state, which represents Africans or prehistoric humans who were traveling at that point of time, if they are similar, right? The time point that can be calculated from the root to this one of the internal nodes. This sort of tells you exactly when during the lifetime of Earth did humans start to actually move from different locations, and that's how you get these kinds of maps. And the last location, which is about 10,000 years ago, they find that humans which are found in South America were the last humans to have sort of colonized Earth based on the time point at which the clustering that they seem to be showing with respect to all the other with respect to all the other human beings.
You said what did it mean by last humans to colonize?
So if you take the humans were traveling, right? So
Yes, sir.
let's say there was some set of humans which started from Africa, right? So basically, so there was these many tribes in Africa that were living, right? Now they cluster separately, right? And the differences between them seem to be much further millions of years ago, right? So for example, these two tribes have split, or so let us say this tribe and this other tribe has split millions of years ago at this point of time, right? Based on the root of this tree. Now, if you go down from this tribe, this tribe basically has two parts. One part which stayed back in Africa, and this other part which is looks very similar to looks to very similar to people who have colonized or which have basically gone to other parts of the world, right? Because all these other people, Japanese, Siberian Inuits, right? Papua New Guinean coast people, Asian Indians, Chinese, they are all clustering together, and they are much different, right, from the people from this Africa. So they are much more similar to these particular Africans, but they are very different from these other African tribes like Igbo, Efik, right? All these tribes which are found here. So now the assumption is that at this root, right, at this time point, the people basically these many million years ago, they would have traveled out of Africa. So we know that these tribes are still existing. So these tribes are related to these people who would have made out of Africa depending upon the mutations that they have these many million years ago, and therefore they sort of have the founding population of these other people, right? But now if you go down to the subtrees, you see that Asian Indians like us, we very early on we diverged from these people, right? So that's why you see that India was sort of colonized around 70,000 years ago by these people. This Saudi Arabia, Middle East was about 100,000 years ago, right? So that is based on the time at which the Asian Indians come up, right? Similarly, Chinese, Koreans, they also seem to have around this area, seem to have colonized around the same area. But now, if you look at people in New Zealand, so these are Samoans, right? So this is 1500 years ago. So actually, these are the newest ones. So if you go down, these ones, Warao and Warao, these are two groups of people. If you see the tree, it's the shortest length from the root to this ancestral node. So these people were the latest people who had sort of colonized. I mean, this is their age of these people, the mutations that they carry. They suggest that their ancestors were only these many years old with respect to all the other people. Similarly, you see this people similar things in Australia as well. You see in Australia, people
Yes, sir. Sir.
colonize.
Yes, sir. Just one
Similarly,
question.
yeah, similarly, people came into North America. So in North America, you have Siberian Inuits, for example. So in Siberia, you have, I think it's not given here. Yeah, I think most of them are relatives of Siberian Inuits. So these are the people who basically crossed Siberia here from Russia into Alaska, and they went down along this coast. Along this is California. Along they went along the coast of California, and the last people who have been found. So you will see that they will have some more small subtrees which are this height, which will be from people of South America.
Yes, sir. Yes. Yes.
Is it slightly more clearer or?
No, sir, it is clear, completely clear. Sir, like even in India, like I can see that in India people come 70,000 years ago. So, sir,
Yes.
after that, in India also people may have mutations and like evolution
Yes.
has not may not have been stopped
Yeah,
70,000
yeah,
years.
yeah, yeah, yeah. But all the people you can basically find that if you try to recreate that their ancestral state, right? With respect to all the other humans of the world, you will see that they will be falling in this tree only, in this part of the tree. They will not be falling in this part of the tree. And that is why we say that Africans have the most diversity amongst all the humans in the world. So that's why we suggest that they are the founding population because there are many, many more mutations existing than can be explained by random mutations or this kinds of evolutionary patterns where one population gets isolated and then they start to, stay in that region.
Yes, sir. So, so genetically, if we see, then South American people are more similar to each other than like African people to each other.
Yes,
Okay,
yes,
understood.
definitely. So now the other thing that I was saying about the beta globin gene, right? So this was like a hemoglobin gene. So there's a specific part of hemoglobin called beta globin. So now if you take about the look at the ancestry, if we took a look at the beta globin, then this is the kind of ancestry that we're getting. We are more similar to chimpanzees than we are to gorillas, right? But if you talk about dopamine, D4 receptor, so this is the receptor in the brain which tells us signals about happiness. So this in this case, we are actually much more different from gorillas and chimpanzees, right? So this suggests that blood or this beta globin based functions are much more ancestral in us than our brain based functions. And this is what allows researchers to say that earlier chimpanzees and gorillas, they had almost all the raw material to make humans, but the biggest change that happened between these primates, these monkeys and us was the fact that we had very different kind of genes in our brain, which led to the increase in the size of the brain. And if you go deeper into what these genes are, what these genes are doing, what kind of mutations they are having, such as there is a mutation called FOXP1 in the FOXP1 gene, which was specifically seen in humans, which allows us to speak more much better than gorillas or chimpanzees. And people have tried to do similar mutations, the same mutations in gorillas and chimpanzees, and they have tried to, these you have seen that cognition has increased, the brain function has increased. So similarly, we can go back to millions of years, even within the mammals which are around us, and we see that humans and mouse are actually much more similar to than what humans are similar to dogs, cows, or whales. So that's why typically a lot of experiments that are happening inside labs are based on mouse, because mouse uh, is much more similar to humans in terms of its brain, in terms of its lots of other characteristics than cows or dogs. And also you will see that there is a very common saying in the West that whales are nothing but cows of the sea, and that actually you can see based on their genetics. Similarly, you can use these things to fight crime as well, because let us say you have in a crime scene, you isolate DNA samples from the crime scene, and then when you match it with the victim, or you match it with some parts on the victim, then you can essentially now make such trees, and you can say that most likely this particular person was their blood was similar to what was found on the crime scene. So I mean, I don't know, probably CID and all these TV serials are not popular nowadays amongst you people, but like if you definitely see something like Crime Patrol, you might be seeing something like this in the background, which is telling you how they are able to match the DNA from a sample of crime. Right. So these are the different implementations and the uses of phylogenetic trees. There are many more that you would encounter, but remember that all of them are based on this very basic idea of how nucleotides and how then amino acids that are encoded by those nucleotides um, are changing between different organisms. All right. So I will stop here, and I will move to the next set of algorithms. I'll take a break and ask if there are any questions before I move to the next topic, which is heuristic algorithms. I want to cover some parts of it today.
Sir, how do we trace time in these trees? Is there a fixed rate for mutation, for random mutation?
Yes. So you assume that mutations happen at a fixed rate. So you know that the error during the replication, right? When the DNA is replicating from one cell to the other cell, you accumulate, uh, say, one error with every billion bases. Okay. So now so every time the cell divides, now you assume that from the day you are born to the day you get sexual maturity, it's approximately 15 years, right? 15 to 18 years. So around that time, how many divisions would your cells have undergone? All right.
Yes,
Now,
sir.
based on that, you can estimate how many million years is it that you would basically certain number of mutations would be like number of years required to actually gather those many mutations. So here we are talking about millions of years, right? So basically gathering one or two mutations in a lifetime is like you only can gather one or two mutations per lifetime. Species, and you look at their evolutionary records, right? And now you compare the mutations that you have between species. So let us say you have species A and species B, and you, based on their physical characteristics and other known fossil records, you have some time between these animals, right? So you say species A and species B were found, they were not found after, say, the subsoil, right? When you go deep under the earth, you say after this layer, you don't find any fossils of this particular species. So therefore, this species must be only this much age, right? Now they have many such benchmark species, right? And they now compare the genes of these species. So you have two ways. One way is to calculate the rate of mutation change based on the mechanics of DNA replication, as I was telling you. So you'll get one rate based on that. And then there is based the number of mutations between species of certain ages, right? That are already known. So based on that, you say, okay, 100 mutations takes 10 million years to happen. All right. So based on this kind of a scale, you can then sort of have that kind of a scale. Do I make sense?
Yes, sir, I got it.
Yeah. So either you actually estimate based on the mechanics of DNA replication, or you compare two known species with their ages known, and you can use that as a calibration. And then given the number of mutations, this can be extrapolated for the age of the ancestor to the leaves. Any other
Okay, sir.
questions?
Sir, I have a question about the parsimony tree. So
Yes.
if at the root more than one nucleotide has the minimum score, so
Yes.
are both those trees equally likely, or there is some criteria by which we decide?
All the trees are equally likely. But the criteria that we decide is based on backtracking, as I told you.
If the roots are like the roots have the same minimum score,
Yes.
then the trees will be different for the.
So can you give me an example, like actually tell me what you mean by looking at this tree?
So like at the topmost point, let's
So,
see.
so first thing is it,
See-
first thing it's clear, right? First we go from the bottom to the top, right?
Yes, sir.
How we are assigning top as 3, this is not a problem, right? This you agree with.
Yes.
This
No,
thing
sir.
you can
And
do.
that if the let's assume that G is also 3,
Okay.
so then it can be either C or either G.
Yes.
Then the trees will be, we can have two equally likely possibilities of trees, right?
Yes. Yes. You can have two equally likely possibility of trees. Yes.
That's what.
So these are called degenerate trees. Like just like you have degenerate solutions to your mathematical equations, right? So these are called degenerate trees. So this is not a yeah, absolute answer. You can get two answers to get two trees. You can get multiple trees also. But then once you do that, once you get multiple trees, then you try to add more and more kind of information and more try to prioritize. Okay, this particular region is more important. This particular region is less important. And then you try to sort of converge to one tree particularly. Any other question?
Sir, how is it related to like in between which we are getting these sequences? How is it related to the characters as we like started with in when we are starting this?
How is it related to character?
Yes, sir.
It is not really related to characters. So characters is a separate thing when we talk about wings and physical characteristics. So now you see, once you have made the genetic tree, now you see you overlay the character tree on top of it. So, so if you, yeah, when you lay the character tree on top of the tree based on the tree made from genes, then you see whether the particular characters were evolved, how many times they were evolved, how many times the physical characters were evolved. Because characters are not passed on, no. I mean, it like if you have your nose in a certain way, giving you an example, okay, your parents are not picking their nose out and giving you their nose, right?
Yes,
Right?
that is correct.
They are they are giving you their genes. Now, given that you have certain genes, your nose might take a certain shape, but if you from the beginning of your life, you're always like holding your nose, right? And you're pinching it, it's possible that your nose takes that particular shape. It doesn't mean that it's only because of genetics. It's sometimes these kinds of characteristics also come due to environment, but the major physical characteristics like number of legs, right? Or whether you will have wings or not, these cannot be environmentally influenced. These definitely have some genetic component to it. So now what you do is, now you see, let us say if we go back to this wing and wingless wala thing, right? So here you see that there were 7 times, 1, 2, 3, 4, 5, 6, 7 times that wings have emerged.
This I'm understanding, sir. If you go a
Listen,
few slides ahead, that is
listen. Let me complete and then you can. So now that basically means that all these species, all these ancestral species, right? All of them, all these particular species, these ones should have something common in their genetics, right?
yes, sir.
Which
Yes.
allows them to have this kinds of have the genes which are responsible for wings. So now you basically go down and down and down and you try to compare, okay, which regions for these ancestral states are not changing. They must be responsible for wings. And you compare it with these other wingless creatures, right? And you compare their genes and you say, okay, now these wingless creatures don't have wings, we know. So what is happening there? So in order to compare physical characteristics with genetic characteristics, you need to have both the trees. You have to overlay this information on top of each other. And this is called the tree reconciliation problem. It's still an unsolved problem in uh, bioinformatics or computational biology, if you say. We were trying to solve this problem with one of your seniors in IIT. Ishank Agarwal, he was in a BTP project, he was trying to solve this problem.
Okay, sir. Okay. Okay, so sir.
Any,
Yes, sir. Can you go ahead two slides more? One more, perhaps. Just one more. Okay. One more. One more, maybe. I don't know. There was a matrix and that was assigned for characters N and M.
yeah, here, no?
Yes. So how can we assign this? Yes. So how can this can be a character matrix? This matrix, how is this made? This I'm not understanding.
This is alignment, sequence alignment only. So I was just showing you an example. Earlier, people were making these phylogenetic trees based on legs and wings and physical characteristics. So they're also characters, right? Yes, having a wing or not having a wing, that's a character. Having 6 legs or having 20 legs, that's a character. Similarly, for each column, you say at this column, do you have a character A or you have a character T? At this column, do you have a character C or a character T?
Correct, sir. Yes. Yes, sir. Understood.
Okay, any other questions? All right. So I'll quickly move to the heuristic algorithms. So far, the kind of algorithms that we have looked at are sort of based on principles that you are gaining out of the sequence itself. They are, you are talking about strings, you're talking about substrings, you're talking about scoring them, but and in most cases you are trying to look for exact solutions of the problem, right? You're trying to optimize to get the exact solution. But now when we go from comparing one sequence to two sequences to multiple sequences to millions of sequences, these exact solutions don't work. As we saw in the case of multiple sequence alignment as well, where we were going for suboptimal solutions, right? So another class of these suboptimal solutions is called heuristic algorithms, which are based on experience, all right, which are based on some fundamental experiential learning. So why is it that we would want to actually look for first place a biological sequence amongst millions of biological sequences? All right. So these are questions that we want to find. Why do we need to find a biological sequence? Right? What do you need? Why do you need a new algorithm for this? And I'll then explain what's a heuristic approach. Then using this heuristic approach definition, I'm going to explain how does the basic local alignment search tool or BLAST works. And then we are going to get into some general principles about how do you score an alignment and how can you say that one alignment is better than the other alignment. and then I'm going to talk about some variations of the BLAST. So most likely this is going to happen. Most of it's going to happen in the next class. So we know that a lot of information in biology is stored in the form of ATGCs, and this information is increasing by the day. So we've already crossed this area where the Moore's law, I mean, we already, if you compare the number of transistors or the speed of computers from 2015 to up to now, it hasn't changed that much, but the cost of DNA sequencing has reduced dramatically, and you have many more sequences, many more gene entries are being added, many more whole genome sequences have been added because of technologies, right? And this has actually led to a real big revolution of big data that is driven by genomic data. So you can imagine that there are about 500,000 like living organisms on average have about 500,000 bases, right? And there are millions of bacteria, millions of fungi, millions of plants, not millions of plant, and all the other species. All of them have that much information. So you can imagine how much information there is there, right? Then people are also trying to find out whether organisms can grow in outer space, how it would look like. And now uh, from the past, I think 5 or 6 years, DNA is also being proposed to be used as a storage device because you can store much more data in DNA than you can do on computer chips. So for example, the entire Wikipedia can be stored in a small 10 microliter volume of DNA. And this is much more efficient than storing it in, say, a USB stick, which is going to take much more uh, space. And you're also having things like people, once you have all this information, people are also trying to create new DNA with synthetic DNA, which is being synthesized inside the lab. All right. So we are in the golden era of genomics where lot companies are also coming up. So I hope that many of you do actually get hired by one of these companies. So for example, Genentech is a big company in this field. Calico is started by founders of Google. Sophia Genetics is a big company in Europe, which is doing these kinds of personalized genomics things where they are using person's own genetic information, their genome, their RNA molecules, their protein molecules to predict their health, right? So there is basically not just 1 billion humans who will be sequenced soon. We also have about 1 trillion cells in our body, and there are about 10 to 100 trillion cells of different bacteria that covers our body. This is called the meta genome. So we have our own genome, and the genome of all the organisms that reside on us forms what's called the metagenome, and that has about 100 times more DNA than human DNA, right? So all this basically means that you have lots and lots of sequences, right? So we have a large database of sequences, approximately 10 to the power 30 nucleotides big. So if we try to do even simple algorithms where we would like to say align, we want to find a local alignment of a sequence to this database, it's going to take a very long time. All right. So typically you would have a query about 1,000 to 10,000 bases long, and you would want to find this query inside this huge database. The algorithms that I've told you so far, they're going to take a very long time, right? So there are these algorithms that I've talked to you about. We have talked about randomized search. We have talked about greedy algorithms. We have talked about dynamic programming algorithms. So the idea is, can we think of something else here? All right. Can you tell me, can we think of something else to solve this problem much faster?
So what is the problem? Can you again?
So the problem is that you have to look for a small query, 100 to 10,000 bases in length, in a large database of 10 to the power 30 sequences. So this is the length of the other sequence. This is the length of your sequence. You want to search this sequence amongst this sequence. So if you can't think of something, I can tell you, I can ask you to look at this, the dot matcher plot.
Yes, sir.
Can you now think of some things? So let us say one of the axis of the dot matcher plot is the database, and the other axis is your query. So does looking at the dot matrix give you some kind of information or some ideas that you could use?
Yes, sir. Wherever the length, the longest length of line segment we will get, that will be the sequence. But again, this dot matcher is also like aligning each and every pair. Then only it is getting that line. Otherwise, it is not getting that line.
Yes. So, but in dot
Then how
matcher,
can it help?
yeah, in dot matcher, there was something called Kamer, right? Right?
Yes,
Did you
yes.
read the how dot matcher works? So it's essentially what it is like in this case, I am plotting each base here, but I'm very well can actually break my query into smaller Kamers and I can plot Kamers, right? Right? So what you suggest is good. We'll find the alignment of our query whenever we have like this kind of a area where there is continuous dots coming up parallel, right? In a parallel location. So those areas will be most likely places where we will find our alignment. And I'll show you how we can actually get to that. So can we simplify the problem, right? And can I tell you how would you search for a sentence in a book, right? Let's say this entire information is stored in a library and I specifically ask you to find the sentence Barrett Joseph climbed a wall in a book. How would you solve? How would you look for this particular sentence in a book?
So first I will look at Barrett. If I will find Barrett, then I will look at if there is a Joseph also. If no Joseph, then go ahead and find the Barrett out. And if
So
I'll
how
find
will you
Barrett
find
Joseph,
Barrett? So in a book, would you go from the beginning of the book till the end of the book trying to find the word Barrett?
yes sir, that I will have to do.
Anybody else would have a better way of finding the word Barrett Joseph inside the book?
Perhaps you can check like in between somewhere, or you can check the table of contents if they are is available. Uh,
So there is something called the index, right?
yes.
So each book has an index. So given you have an index, you can actually see that this particular word Barrett Joseph is given in these pages of the book. So we will go to these pages, right? And then we will do exactly what you were saying. We will go to the right side of Barrett Joseph and see whether the words climbed a wall were following Barrett Joseph. All right. So the algorithm that is the basis for BLAST and these heuristic algorithms, these kind of algorithms is to first build an index of your database. This is typically an index made of Kamers, right? We know what a Kamer is. Kamer are substrings of the index, and you essentially create the index. You tell where all those Kamers are lying, and once you search for the same Kamers in your query sequence, right? You try to basically extract Kamers from your query sequence, and you see where in the index you go to the index of your database, looking for those Kamers, location of those Kamers, and then you search and extend. So can you tell me what kind of alignment would this be? Would this be a global alignment or a local alignment?
This will be local alignment.
Okay. Ashi Dev, Deepak, global, local.
Sir, globe. I have no idea.
So, not really sure about it.
You are looking for local similarity, right?
Right, sir. So it would be local alignment itself.
Local alignment. So this is what a heuristic approach is. I'll just close here. It's based on prior experience. Prior experience, basically, it's not mathematically precise. The prior experience says that if you find a few Kamers of your query in the database, then most likely your alignment is also going to be starting from there. Okay, so this is based on trial of error, process of elimination, right? So when you are searching a book or aligning a sequence, your experiences, if there will be an alignment, then the two aligned, the location where the alignment is happening and your sequence is going to share Kamers. All right. So in the next class, I'm going to show you how this algorithm works and it's and how do you score an alignment when you do this algorithm and a few variations of this algorithm. And after that, I think we'll have um, the lab will also be based on BLAST searching. All right, so I'll stop the class here. I'll take any questions if you have. Otherwise, you're free to leave for the
Yes,
next.
sir. Yes, sir. Sir,